Immunology Insights, Basics and Beyond, Chapter 59, Next Generation Checkpoint Therapies and Combination Immunotherapy. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our previous chapter, we explored monoclonal antibodies, the workhorses of modern oncology, and how they block tumor growth, recruit immune killers, or ferry toxic payloads straight into cancer cells. Today, we are staying within the world of immunotherapy, but we shift gears, away from antibodies that attack tumors directly and towards therapies that liberate the immune system itself. We've already met the major checkpoint pathways, CTLA-4 and PD-1, and the scientists behind them in earlier episodes. Those therapies were a revolution, but as powerful as they also revealed something important. Tumors don't rely on a single brake pedal. They install entire braking systems, redundant inhibitory circuits designed to exhaust, mislead, and paralyze T cells. In this chapter, we dive into next-generation checkpoint therapies, the new inhibitory receptors under clinical investigation, and the logic behind combination therapy, where multiple immune pathways are manipulated at the same time to produce deeper and more durable responses. So let's talk science. But why do we need actually new checkpoint therapies? PD-1 or programmed DES1 and CTLA-4 or cytotoxic T lymphocyte-associated protein 4 inhibitors transformed oncology. Melanoma, lung cancer, renal cell carcinoma, Hodgkin lymphoma. Diseases once notorious for poor outcomes suddenly became treatable in entirely new ways. But the clinical truth is sobering. Only about 20 to 40% of patients respond to single-agent checkpoint blockade, depending on the tumor type, and many of those initial responses are not durable. T cells in the tumor microenvironment do not only express one inhibitory receptor. Chronically stimulated or exhausted T cells often co-express several brakes at once, such as LAG3, TIM3, TIGIT, and others. Blocking PD-1 alone is therefore like cutting one wire when the tumor has installed an entire panel of safety systems. Next-generation checkpoint inhibitors aim to target these additional pathways, either alone or in combination with the classical PD-1 CTLA-4 blockade, to restore T-cell function more completely and achieve deeper, more sustained remissions. Now, let's look in detail at the respective new checkpoint inhibitors. First, we have LAG3, the first next-generation checkpoint in the clinic. LAG3, or lymphocyte antigen 3, is expressed on repeatedly stimulated or chronically activated T cells. When engaged, it dampens T cell receptor signaling, reduces cytokine production, and limits proliferation. Its main ligand is MHC class II, and binding transmits an inhibitory signal distinct from but complementary to PD-1. In many tumors, PD-1 and LAG3 are co-expressed on exhausted T cells. Blocking them together can reinvigorate cytotoxic activity more effectively than targeting either alone. Clinically, this was validated in melanoma, where the combination of the PD-1 antibody nivolumab with the LAG3 antibody relatlimab improved progression-free survival compared with PD-1 inhibition alone. The effect size was not dramatic, but it was a milestone, the first proof in humans that targeting an additional checkpoint beyond PD-1 and CTLA-4 can meaningfully enhance response. Second, we have TIM3, an exhaustion marker with a dual role. TIM3, or T-cell immunoglobulin and mucin domain containing 3, is another key exhaustion receptor. It is found on chronically stimulated CD8-positive T cells, but also on regulatory T cells, dendritic cells, and some myeloid cells. Its ligands include galectin-9, phosphatidylserine, and CEACAM1, which transmit slightly different immunoregulatory signals depending on context. In early immune responses, TIM3 can have activating or modulatory functions. In chronic settings such as cancer or persistent viral infection, however, it becomes predominantly inhibitory and contributes to the exhausted phenotype. Many tumors upregulate TIM3 on T cells, especially after PD-1 blockade, using it as a backup brake. Therapeutically, TIM3 blocking antibodies are in phase 1 and 2 trials. As monotherapy, they have shown limited activity so far, which fits with their biology. TIM3 is part of a network of brakes, not the only one. The most promising data comes from combinations with PD-1 blockade, where co-inhibition of both receptors further restores T cell function in some patients. Third, we have TIGIT, connecting T cells and NK cells. TIGIT, or T cell immunoreceptor with Ig and ITIM domain, sits at the interface between adaptive and innate immunity. On T cells, it inhibits activation and cytokine secretion. On NK cells, or natural killer cells, it reduces cytotoxicity. Its main ligand, CD155, is widely expressed on tumor cells and antigen-presenting cells. Because TIGIT and PD-1 are often co-expressed on tumor-infiltrating lymphocytes, dual blockade was expected to be strongly synergistic. Early clinical data in lung cancer indeed showed encouraging response rates when anti-TIGIT antibodies were combined with anti-PD-1, PD-L1 therapy. However, several large phase 3 trials later failed to meet the primary endpoints. This does not mean that the TIGIT pathway is irrelevant. It more likely indicates that we need better biomarker selection, for example identifying tumors with high TIGIT CD155 signaling, improving antibody formats and optimizing combination partners and schedules. Biologically, TIGIT remains an attractive target because it connects T cells and NK cell inhibition. Clinically, its role is still being defined. And fourth, we have VISTA, targeting the myeloid compartment. While PD-1, LAG-3 and TIGIT are mostly discussed on T cells, VISTA, or a V-domain immunoglobulin suppressor of T cell activation, stands out because it is highly expressed on myeloid cells such as macrophages, dendritic cells, and myeloid-derived suppressor cells, or MDSCs. Instead of directly inhibiting T cells, VISTA helps establish an immunosuppressive tumor microenvironment. VISTA expression is particularly prominent in tumors with dense myeloid infiltrates, such as some prostate and pancreatic cancers. In preclinical models, blocking VISTA can reprogram the microenvironment towards a more pro-inflammatory state, decreasing suppressive myeloid activity and allowing T cells and NK cells to regain traction. Early phase clinical trials of VISTA targeting agents are underway and represent a conceptual shift, not only releasing T cell brakes, but also re-educating the suppressive myeloid compartment. So why is combination therapy so important? If first-generation checkpoint inhibitors were like flipping a light switch, modern immunotherapy is more like adjusting a complex mixing console. Tumors use multiple inhibitory receptors, physical barriers, metabolic suppression, myeloid-driven deviation, and regulatory T cell recruitment to keep immunity at bay. It is therefore unlikely that blocking a single pathway will be sufficient in most advanced cancers. Combination immunotherapy tries to address this complexity. One approach is to pair PD-1 blockade with a second checkpoint such as LAG-3, TIM3 or TIGIT, targeting primary and secondary brakes simultaneously. Another strategy is to combine checkpoint inhibitors with tumor-directed monoclonal antibodies. As antibodies kill or stress tumor cells, they release antigens and danger signals, which reinvigorated T cells under checkpoint blockade can recognize more efficiently. Combinations with cell-based therapies are also being explored. CAR T cells and TCR-engineered T cells can become exhausted in a hostile microenvironment of solid tumors. Adding PD-1 or LAG-3 blockade or engineering CAR T cells to be intrinsically resistant to these checkpoints may prolong their activity. Likewise, pairing checkpoint inhibitors with therapeutic cancer vaccines aims to generate new waves of tumor-specific T cells and then keep them alive and functional once they infiltrate the tumor. Myeloid-targeted approaches such as VISTA blockade can be layered on top of T-cell checkpoints, especially in tumors where suppressive macrophages and MDSCs dominate. The overarching idea is simple but powerful. No single immune lever is enough, but thoughtfully combining several can shift the overall balance in favor of tumor control. However, some tumors still resist. Why is that so? Even sophisticated combinations run into significant obstacles. Some tumors have very low antigenicity, presenting too few recognizable neoantigens for T cells to attack. Others are physically shielded by dense fibrotic stroma and abnormal vasculature that exclude immune cells. Pancreatic cancer is a classical example here. Many create extreme metabolic stress zone with hypoxia, acidosis, and nutrient depletion. Under these conditions, T cells remain dysfunctional even if checkpoints are blocked. Dominant suppression by myeloid cells and regulatory T cells can further blunt any attempted immune reactivation. In such settings, simply adding more checkpoint inhibitors risks toxicity without much benefit. For these cancers, next-generation strategies integrate checkpoint blockade with additional tools, agents that normalize tumor vessels or remodel stroma, metabolic modulators that relieve nutrient and oxygen stress, oncolytic viruses that inflame cold tumors, and locally delivered cytokines or drugs that concentrate immune activation directly in the tumor. Checkpoint therapy becomes one component within a broader immunological ecosystem rather than a standalone solution. As a future outlook, we need to precisely tune immune activation. Looking ahead, next-generation checkpoint therapies are unlikely to replace PD-1 and CTLA-4 inhibitors. Instead, they will build on them to refine and personalize immune activation. Tumor immune profiling will increasingly guide which checkpoints to target in which combinations, based on patterns of inhibitory receptor expression, myeloid infiltration, and T-cell exhaustion signatures. We can expect more bispecific or multispecific antibodies that block two inhibitory receptors at once, Fc-engineered antibodies with tailored effector functions, and delivery strategies that concentrate immunomodulators within the tumor. For example, via intratumoral injections or slow-release depots to reduce systemic toxicity. Checkpoint blockade will be combined more systemically with vaccines, cell therapies, targeted agents, and microenvironment modifying drugs. The trend is towards subtler, context-aware manipulation of the immune system, not simply turning immunity on, but dialing specific pathways up or down for each patient and each tumor type, aiming for deep and durable tumor control without overwhelming autoimmunity. To conclude, next-generation checkpoint therapies build on the foundation laid by PD-1 and CTLA-4 inhibitors. They recognize that tumors suppress immunity through multiple redundant pathways, and that restoring effective T-cell activity requires multi-layered intervention. By targeting inhibitory receptors like LAG3, TIM3, TIGIT, and VISTA, and by combining checkpoint blockade with monoclonal antibodies, cell therapies, vaccines, and microenvironment-focused agents, modern immunotherapy is becoming more adaptive, more nuanced, and more powerful. In our next chapter, we will extend this journey even further, exploring adoptive cell transfer therapies beyond CAR T-cell, TCR therapies, tumor-infiltrating lymphocytes or TIL therapy, and next-generation NK cell treatments and more. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So see you next time, and thank you for listening.